Welcome back. Here we go for our last part 4, part D of our chemistry chapter. What on earth do a cheeseburger, a sunflower, and a cute baby pig have in common other than that they're all delicious? Sorry, actually, they're all made of the same stuff, the same macromolecules. You can see it most easily right over here in the cheeseburger. Protein, fats, and carbohydrates. Of course, these are all living things, or were once living things, and so they still contain DNA. So there we go. That's what organic chemistry is really all about. Those four so-called macromolecules: carbohydrates, lipids, proteins, and nucleic acids. We tend to focus on these ones, DNA and RNA, but let us not forget that ATP is also technically a nucleic acid. So these are the four macromolecules. Remember that they're all synthesized. They're all made the same way using a chemical process called, do you remember? Dehydration synthesis. All of these macromolecules are disassembled the same way too, using a chemical process called hydrolysis. And if you forgot, you might want to go back to the last video. Here we get a little bit more of the details. How do we build these carbohydrates? Well, we start with these monosaccharides. We get most of these from our diet. We can technically make some of these, and we'll learn about that much later in our semester. But in reality, we get our monosaccharides from diet: sugar, starches, French fries, potatoes, wheat, chips, rice, pasta, right? Carbs. They are monosaccharides, one sugar. Monosaccharides like fructose, glucose, and galactose. Now, we can synthesize disaccharides by combining glucose and fructose into sucrose. This is table sugar. This is what we find in high fructose corn syrup and in our frostings and things like that. Sugar is sucrose. Although we like all this stuff, it all tastes sweet to us, as humans, I mean. Now, if we want to store up a carbohydrate, we want to build those simple sugars, 1, 2, 3, 4, 5, into a big unit called a polysaccharide. Many sugars. Polysaccharides are starches, right? Things like amylose and glycogen and cellulose, which we find in plants. So it's easiest for us to eat plants, and then we just convert these starches into blood sugar, monosaccharides, and then through dehydration synthesis, we hook those together into polysaccharides so we can store them for short-term energy usage. The next macromolecule we're going to talk about are lipids. And although there are other lipids like cholesterols and some derivatives of that, most of the time in this class when we talk about lipids, we're going to be talking about triglycerides. Sometimes we talk about phospholipids, and sometimes we talk about cholesterol and steroids. But look at this. We'll call triglycerides our poster children for fats or lipids. They really do a nice job of representing it. Look at this. A triglyceride has 1, 2, 3 fatty acids covalently bonded here to a glycerol molecule. And so despite the fact that we do have a couple of little regions where you might think there is a little bit of polarity, this is a non-polar molecule overall. And that's what we see here, the triglyceride, the finished product. After dehydration synthesis, we have a triglyceride. So what are the reactants? Glycerol and 1, 2, 3 fatty acids. What's the product? The triglyceride molecule. And you can see the functions in the human body include protection, like cushioning, insulation for thermal, and as an energy source. This happens to be nature's long-term energy storage molecule, triglycerides. And so if I gain weight, I am storing it as triglycerides, fat. We'll talk a lot more about the specific types of fats in our digestive and metabolism chapter coming up. But for right now, we just need to understand these three: saturated fats, unsaturated fats, and trans fats. We'll start at the bottom. Trans fats are totally artificial. We don't find them in nature. And so, like so many other chemicals, these totally unnatural fats have nasty health effects. And highest cardiovascular risk fat, right? So hydrogenated is the word you want to look for. Even if it says no trans fats, if it has hydrogenated fats in it, it does have trans fats. And we'll talk about how that deceptive labeling is hurting people a little bit later in the semester. Next, in terms of health effects, is saturated fats. These are not good, especially if they're animal-based. Saturated meaning that they have all of the hydrogens that you could pack in there. Unsaturated means, well, technically, if we could change these double bonds into single bonds, I could fit a hydrogen here and a hydrogen here. And so this is not totally hydrogenated or totally saturated. Saturated fat has all of the hydrogen. And then those trans fats, they pack even more and change it in a way that it becomes more disease-provoking or pathogenic. Here is another type of lipid, not a triglyceride, but a phospholipid. Really important. We'll talk more about it later in the chapters, but this one is one that forms a part of the cell membrane. And so we can see this phospholipid means a phosphorus hooked to a fat. And it comes in this form: the phosphorus head, which likes water, and the fatty acid tails, which don't like water. And those characteristics totally determine how the cell membrane acts. And we'll talk about that in the next chapter. Our next macromolecule, proteins. These are long chains of amino acids. They're formed through dehydration synthesis into a special super strong covalent bond called a peptide bond. So don't be confused. This isn't a new bond. It's a covalent bond. It's just a super strong one because of the nitrogen here. So peptide bond forms a dipeptide. If we take 1, 2, 3, 4, 5, 6, 7, 8, 100 amino acids, we can hook them together into a long chain called a protein. I got red amino acids and blue ones and orange ones and purple ones and green ones. And it turns out that the pattern of the amino acids will determine the location of the hydrogen bonds. Remember, these ones allow the protein to fold into a three-dimensional shape. And when it comes to proteins, it is all about the shape. This is hemoglobin. And look at the complicated three-dimensional shape that we see right here. If it were even a tiny bit different, it wouldn't carry oxygen as well. And that's just one of hundreds of examples we might have a chance to talk about. Don't forget that these enzymes, biological catalysts, are also proteins. And so, of course, the shape is super important. The shape of this enzyme, sort of like a little mustache, fits these two molecules. And by binding to both of them, it lowers the activation energy, allowing them to combine to form a new product. And then it goes back to a mustache. So look at this. The mustache can work many, many times to bind more molecules together. And that's how it speeds up these chemical reactions. That's why enzymes are catalysts. Now, the issue is the location of these hydrogen bonds, these little red dotted lines, determines how it folds and in what shape the enzyme will fit. So if we change the temperature, or if you have a genetic mutation, or if you're suffering from acidosis, your enzyme won't be in the right shape, and then that's going to lead to some sort of a disease process or a metabolic problem. Great video. I don't want to waste your time on this video. You can watch this one whenever you would like, but you're never going to think about Jell-O the same way again. I promise you that. And it talks about how proteins can be misshapen by changes in their environment. Just a couple more quick things and then we'll be done with Chapter 2. Nucleic acids, DNA, RNA, and ATP. Now the next chapters talk all about DNA, so I don't even want to go into this too much aside from just giving you some basic vocabulary terms. You know that DNA is your blueprint for life, right? Your genetic code. Did you know that your DNA is comprised of only 4 little chemicals? And that it is the order of these chemicals that spells out the recipe for your unique proteins, right? So in the same way that I can spell lots of words with the 26 letters in our alphabet, biology can spell out lots of proteins with just 4 of these individual nucleotides. Cytosine is a nucleotide. Guanine is a nucleotide. Thymine and adenine, these are the building blocks of DNA. And then RNA is sort of a protein assembly machine we'll learn all about in the next chapter. RNA uses cytosine, guanine, adenine, and uracil, not thymine. When we combine them in DNA anyway, the T's, the thymines, always hydrogen bond with adenines, and the guanines with cytosines. And it is these hydrogen bonds which give DNA its unique shape. So look at that. The double helix is formed by those hydrogen bonds, very much in the same way that hydrogen bonds influence the shape of proteins. Last but not least, adenine triphosphate, or ATP. ATP is a rechargeable battery that our cells use as the energy currency within the body. It provides energy for other chemical reactions which need that little energetic boost. And ATP also is the chemical that drives our physical contractions, which allow us to move and speak and swallow and digest and to have a pulse and to have blood pressure. So ATP. Where does the energy in ATP ultimately come from? It's from the energy in our food. Unlike plants, we can't just hang out in the sun and store up energy. We have to eat things, right? Plants and animals give us energy that we convert or store in this energy molecule, which we can then move over here and then harvest the energy to do some work, make something move, or push a chemical pathway. And then what was left is ADP and a little inorganic phosphate. We can feed these 2 chemicals back into the chemistry machine, and using the energy from our food, we hook them back together. So this 3rd phosphate hooks in, and we now have ATP. The battery is recharged. So this just goes around and around. As long as you have oxygen and you are eating food, you'll be all right. Although you don't have to eat continuously, right? I'm just kidding. Just because I do doesn't mean you should. Here we go. This is how ADP is converted into ATP. Notice that we have to put a lot of energy in to make the product. This is an endothermic reaction, right? It requires energy. This happens, by the way, inside of our cells in a tiny creature called a mitochondrion. We'll talk about that in the next chapter. And then now that we have the high energy ATP, we can degrade it through hydrolysis to release the energy for movement or for a heart contraction or to secrete some product. All right, please review the last part of Chapter 2, and I'll see you back for Chapter 3.